Welcome back, everyone. So today's review is going to be going over chapter 9, which has to deal with the genetics of organisms in general, not just microorganisms. So genetics basically is looking into the study of a genome of organisms, and the genome is essentially the sum of the genetic material of an organism, including any of the extra chromosomal DNA. So what does that mean exactly? Well, remember that bacteria can carry those little extra circular bits of DNA that can code for things like antibiotic resistance. Those are called plasmids. So those are included in the genome. So literally anything, or for you in your case, you have genes in your chromosomes that are part of, your chromosomal DNA inside of your nucleus, that code for, your eye color and everything like that we've talked about previously. But you also have genetic information in your mitochondria, because your mitochondria contain their own genome essentially within themselves. So together, all of that, your genome includes your own chromosomal DNA as well as any other extra chromosomal DNA, including mitochondrial DNA. So I did want to make sure we understand that concept. The genome is everything in that organism that is coded for in any sort of genetic material. So what is a chromosome? That's something I keep talking about. Well, basically, a chromosome is a structure that contains the genetic material that codes for the genes that make up that organism and determine basically what that organism's functions and characteristics would be. That's the chromosome. So we know as eukaryotes with true nucleus, we have multiple linear chromosomes, all those X's that we see lined up. It's called karyotyping. That is a depiction of our chromosomes. Whereas prokaryotes, like bacteria, they typically have one circular genome. It's just one big circle, and it's really big. All right. So that brings us up to the concept of genotypes and phenotypes. Essentially, genotype is whatever your genetics are coding for, and your phenotype is what is being expressed. So perhaps your mom gave you the genes for blue eyes, and the dad gave you genes for brown eyes, and you have brown eyes, for example. You might be carrying both of those genes, but only one of them is being expressed. Your genotype is that you have gene for blue and a gene for brown, but your phenotype is what's expressed, and that would be that you have brown eyes. So that is an example there. Between genotype and phenotype. So genotype, literally the genetic makeup, phenotype, what's being expressed. All right, so all of this stuff is encoded within our genomes. We keep talking about our genomes and our genetic material and DNA and all of this. What is DNA? DNA is deoxyribonucleic acid. So it's deoxyribo because it was ribose sugar. We removed one of the oxygen groups essentially. So deoxide it. So it is deoxyribose is the sugar. It is a nucleic acid, just that meaning the type of molecule that it makes up. So that's going to consist of a deoxyribose sugar. It's a 5-carbon sugar minus the oxygen that would be seen in ribose found in RNA. Then we have the nitrogenous base, whatever it might be, the A, T, C, or G. That's the nitrogenous base portion, okay? And then the phosphate group. And then we just repeat that nucleotide, which is that nitrogenous base plus the sugar, plus the phosphate group over and over and over again to make up one side of that double helix. And then on the other side of the double helix, so we just make up one, oh, here it is. So one side of the double helix, so maybe you have an A on one side. We'll just draw a few. I promise it won't take too long. A G on this side, a C on this side, whatever. All right. So each of these is made up of a nucleotide, which is a ribose, a nitrogenous base, which determines A, C, T, or G, and the phosphate group. The phosphates, essentially repeating sugar phosphate backbone. The phosphates give it that negative charge on the outside of the helix. So the other side, we have another strand that matches up where A always binds with T, G always binds with C, okay, always. That's DNA. So this essentially is what makes up the double helix. One side is, you know, what we started out with, and the other side is the opposite, the binding match to it. We would say when we're looking at a gene, one strand is the coding strand and one strand is the template. So one strand, you could read it as it would give this amino acid and this amino acid and this amino acid as a codon. The other one is the opposite that we would use to make the complementary mRNA, messenger RNA. So that's basically how that would work out. Right, so what else do I want to say about that? I'm not really sure. So they all run in a 5' to 3' direction. And so one goes 5' to 3' direction. The other one does as well, but it is anti-parallel, meaning that if this one's going 5' to 3', 5' is always the start, by the way. This one's going 5' to 3', then this one's going 5' to 3' in the other opposite direction. What does 3' and 5' mean? That just means a certain spot on the sugar that is exposed at that end. That's all it means. It's just part of the structure of the sugar that you would see at that end. It's just a numbering thing. So they just numbered the carbons in the sugar, and that's it. That's all that means. So DNA, deoxyribonucleic acid, we have a nitrogenous base, deoxyribose sugar, 5-carbon sugar, and a phosphate group. We have the two anti-parallel strands, which we just introduced that concept of 5' to 3' and 5' to 3' in the opposite direction. And that just refers to the orientation of the nucleotides or those sugar molecules as part of the nucleotides. Replication means making copies of the DNA. So we need to do this whenever we make new cells, right? Anytime we make a new cell, you need to make a copy of your DNA so that they have identical DNA in them. Replication is what we call semi-conservative. So essentially, what that means is get out of the way, because I was going to drop there. Get out of the way. Okay. If this is your original cell's, you know, chromosome or whatever, whenever we replicate it, we are going to split it apart into two strands. Now, this is real watering down, right? Because we know that it's a little bit more complicated than that, because there is so much DNA that it's all wound up in that helix, and we have to unwind it as we go. And we'll move along at different spots in the chromosome. For eukaryotes, we'll pick many spots throughout the chromosomes, and then they'll all just like get sealed together at the end. Whereas in prokaryotes, they kind of have two origins of replication, and then they both go around and then meet each other and then seal together. So that's how that would work, eukaryotes versus prokaryotes. But anyway, so they'll split apart essentially, and this can happen gradually throughout the chromosome because again, it's a large piece of DNA. And then we will make copies A to T to G to C and whatever. So this would be the new strand as we do that. And for this one, we would have, this is the new strand. So now we have a copy. We have two things where we had one to begin with, 2 bits of DNA where we had 1 bit of DNA to begin with. And one of each new DNA chromosome consists of an old piece and a newly synthesized piece, right? So part of it is old, which is the red, and then now we made a new portion, which is the blue here. That is what semi-conservative is referring to. That's what that term refers to. Semi-conservative because part of the old, and we also have some new added that we made for the new strand. Okay, so the double helix splits into 2 singles, and then we make copies that match up with that single. So we now have 2 new double helices that consist of an old strand and a new strand, right? So that's what the old is the red and the blue is the new. So in replication, when we are doing all of this, DNA polymerase always adds nucleotides in the 5' to 3' direction. This creates a problem as we are opening up, because again, we don't have them in like nice tight little packets like this that are shown in this figure. What we have is long stretches of DNA, and then we're going to open it up and unwind it because again, it's like a helix, right? So we're going to open it up, and remember, one strand is going to be 5' and 3', and the other one's anti-parallel, right? So this one, we open it up, and this is 5' to 3'. So as we open these guys up, we will be adding in the complements to these only in the 5' to 3' direction. And that means that as we are opening these up and adding new nucleotides, we are opening these guys up. We can add, again, this one would be 5' in blue. So this is our new strand, right? So we can add it here and then here and then here, and we can just keep going because we started at our 5' and we're adding towards its own 3', right? So we'll just keep adding and adding and adding. That's fine as it's opening up into this direction. However, the other strand, we have to add in 5' to 3'. So we can't add starting from here and then moving on from here to here. No, we can't do that because this has to be the 3', and we can only add 5' to 3'. So what is our solution to this? Well, as we are opening up and we are making a copy of that lead with that leading strand, which is the one on the bottom that's just going and doing its thing just fine, just straight on through. That's the leading strand. We are opening up the lagging strand, and we have to work with that one in pieces. So essentially, what we're going to be doing, we're going to open up. So we've opened up a chunk of it, and now we can add on from 5 over here. We can add on here, 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 and whatever. We can do that little piece of it, right? But then it's going to keep opening up, and we're going to have to keep adding little pieces like that. So we also can't just add nucleotides for DNA on out of the friggin' blue and match up with DNA. We have to add them onto the ends of other pieces of nucleotides. So we use RNA as primers for this. We add a little bit of RNA in the beginning, and we build off of that, and then we will eventually get rid of that RNA and fill it in with DNA later on, and then seal everything together. So as these pieces are made in this other strand, this is the lagging strand, because obviously it's taking a little bit longer because we've got to wait for it to open up, and then we'll add a little piece. Each piece is called an Okazaki fragment. And then once we have our Okazaki fragments made, we can seal them together with DNA ligase. And DNA ligase essentially is just something that we use to seal up pieces of DNA together so that the phosphodiester bond, which you don't really need to worry too much about what that is, but it's just the bonds between the nucleotides. So we seal those up and they're all one piece. You need that to happen in order for us to be able to use those genes later on when we are encoding for whatever gene we might need to be reading. So I think that covered all of that. All right, so RNA is ribonucleic acid, similar concept here, but we're using ribose sugar. So it still has the oxygen on that sugar that we had removed for DNA, the deoxyribose. We use it for a lot of things in the cell. We can use it for, most importantly, I think most commonly what we will be talking about the most would be as messenger RNA. So that's basically making a working copy of a gene. So if your DNA is your main blueprint, and we want to keep that in the nucleus where it's nice and safe. We don't take that out of the archives at the library, right? So we go in there and we make a copy of the thing that we're interested in, the gene that we want to look at, and we make it in RNA form, and that is messenger RNA. It codes for the gene as the working copy of that gene that we're going to take out into the cytoplasmic part of the cell, or in the endoplasmic reticulum, and associate with some ribosomes. And then we're going to read that messenger RNA message and turn it into protein. When we go from DNA and make that RNA copy, that's called transcription. When we go from RNA into protein, we're reading that copy and making our protein product. That's called translation, okay? Both transcription and translation have 3 essential steps. That is initiation, elongation, termination. Pretty straightforward, pretty much exactly what it sounds like, right? We start making our mRNA copy, and then we just go through it, and then we, that's called elongation, keeping on going, and then terminating whenever we get to, the end. Some sort of signal there to tell us that we need to dissociate and we're done. So DNA polymerase makes DNA copies. RNA polymerase makes RNA from the DNA blueprint, okay? The difference also, so we have ribose sugar, that's one difference between DNA and RNA. Another difference between DNA and RNA is that RNA uses uracil instead of thymine, so U instead of T. So that's a big difference as well. So once we get into reading our codons, which are three A, U, G, U, U, A, whatever it is, every three that we have that is known as a triplet in the DNA, a codon in the RNA, and the messenger RNA. Those guys count for one amino acid. And so we read those using uracil, of course, if we're looking at the messenger RNA. For eukaryotes like you and I, we have something called introns that we have to clip out. And then we will join together the exons of the messenger RNAs. Introns, they don't code for anything, and they'll mess up our ability to make our gene. The exons, we'll join them together and then we can make a full gene. This is fine. This helps us control our gene expression as eukaryotes. But if we're trying to put our genes, like the gene for insulin, into a prokaryote to make that protein for us, then we need to have our exons already put together when we put it into the bacteria, because the bacteria can't do that for us. It can't clip out the introns. So that is something to consider about in genetic engineering, which we'll talk about when we move on into the next chapter. So every 3 DNA nucleotides, the A, T, C, G, that's the triplet. In RNA, that refers to the corresponding triple, A, U, G, whatever it is, that is the codon. Codons are recognized by transfer RNAs, tRNAs. They have an anti-codon on them. So, A with U, G with C. And it binds them with that anti-codon. And if that anti-codon on the transfer RNA matches up with the codon on the messenger RNA, then the amino acid associated with that anti-codon on that tRNA will be added onto the next amino acid in the line as we are translating things. And essentially, that's how things are going to move on with translation. We have our messenger RNA. Let's try. We have our messenger RNA. Ribosome comes and associates with it, and we have these little pockets that the tRNA can come into. And tRNA, they look sort of like hairpins that are bent all up, and they're associated with a specific amino acid. And then that amino acid, I don't know, alanine, phenylalanine, thymine, not thymine, sorry, that's a nucleotide. So alanine, phenylalanine, tryptophan, leucine, there's 20 of them. I'm not going to go through all of them, but yeah. So there's all of those that'll be coded for based on those triplets in the messenger RNA matching up. So the codons in the messenger RNA matching up with the anticodons of the tRNA, and then joining the amino acids together as they bind into one another. And the ribosome will move down and add in, this amino acid gets added to the next one as it matches up, and then to the next one. And the ribosome shifts on down and keeps on reading and adding on more amino acids and so on and so forth till our polypeptide is done. And remember, a peptide bond is whenever we are binding, that's what occurs when we are binding our amino acids together. That NH2 group at one end to the carboxylic acid group at the other end of the next amino acid. So that is a peptide bond. That's why we call it a polypeptide, because we have a bunch of those going on, right? So we have our polypeptide, and some proteins, that's all they need is just to be that polypeptide. Other ones have to have different parts, different polypeptides come together and act as a unit together in order to perform their protein function, which we talked about in the chemistry chapter a little bit with quaternary structure of proteins, right? So that's the whole point of all this. We're making proteins, and proteins are doing the work. Proteins are our workhorse in the cell. So in bacteria, in prokaryotes, we have operons. They can be inducible or repressible. In the case of an inducible operon, basically we are, in the presence of lactose, we will start being able to make genes that will allow us to break down lactose. And what happens is we have a repressor that prevents this. We don't want to make our stuff all the time, right? We don't want to be making proteins that are associated with breaking down lactose if there's not any lactose. What's the point in that, right? You're wasting the cell resources. So we have something bound to, the operator basically that keeps the RNA polymerase from making the messenger RNA. And whenever we have lactose come in, it comes in and binds onto that repressor that's preventing all that from happening and induces the activation of the whole operon. In bacteria, they have genes that can be turned on in one big old long stretch. They're all just like related to that one function, but multiple genes related to that one function. It's multiple proteins related to that one function. So that's what happens with the lac operon. It'll be induced by the presence of lactose. The other operon that we talked about was the arginine operon, which was a repressible operon. So it's on. It's going to be making arginine, which is an amino acid. It's going to be manufacturing that amino acid for the cell until there is so much of it that has been produced that it will basically associate with a repressor and cause that repressor to change a shape so that it now can bind back on with the operator, and we shut down manufacturing of those proteins that are associated with manufacturing arginine. So that is how that one would work. So that is going to be repressing the operation of all of that. So repressible operon. Bacteria can also transfer their genes from one bacterium to another. Essentially, this has to happen from the same species to same species, okay? So E. coli isn't going to be transferring necessarily its genes to a totally unrelated bacterium like staph or something. So it has to be species to species. But there are three major ways that you do need to be aware of. That's that they're able to transfer their genes, and that will be through conjugation. It's what it sounds like. They use the sex pilus, which is just a tunnel that forms between two cells that allows for genetic material to move across. You can often copy a plasmid or something, or even part of a chromosome, and just shuttle it through the tunnel, if you will, the pilus, while it's being replicated. And that can happen like in action, if you will. Then we have transformation. Essentially, in transformation, you have competent cells. Those are cells that are able to take up DNA from their surrounding environment. And when they do that, it could be in the form of full chromosomes, pieces of chromosomes, or plasmids. So they will take it up into their own cell and start expressing the genes that way. So it gets incorporated essentially into their whole genome in that manner. And then we have transduction. So this would be transmitting genetic material through infection by bacteriophages and lysogeny. Some bacteriophages will come in, incorporate themselves into the bacterial DNA, and they can pick up just random genes, any gene in the entire bacterial genome. It will come in, incorporate itself into the genome. And then when it comes back out to make more viruses, just take some of that with it, some of the bacterial genome with it. And then it can package up little pieces of the genome of the bacterium as well whenever the bacteria is infected with the virus. So those are some ways that can work. Sometimes when we're just packaging up random genes. Anyways, moving back on to this. So bacteriophages, infecting bacteria, they're viruses that infect bacteria. When they start packaging up their new viruses, can just pick up genes from bits of DNA in the bacteria that it's infected. It can pick up any gene that way. So technically you could be carrying any gene from any bacterium to the next infected bacterium in that manner through infection via bacteriophage. What are you looking at? Anyways, then you can also have it more specific. So some bacteriophages will insert themselves into that DNA in specific spots, predictable spots, if you will. And then that will, be predictable as far as like what gene they would be picking up whenever they come back out of the DNA. So if they always insert themselves into the same spot, they'll always be picking up the same kind of DNA. You can predict essentially what genes they would pick up. So those are the ways that transduction will work. So you have, for horizontal gene transfer, And what that means horizontal, it means between different bacteria. If you inherit a gene from your parents, that's vertical gene transfer. So these bacteria, that's not getting it from like their, parent cells. They are getting it from different cells that they're associated with. So that's horizontal. So conjugation, transformation, transduction. Okay. All right, and then talking about mutations in genetics, essentially, let's just run through them real quick. Anytime that a nucleotide gets changed from what it originally was into another nucleotide for whatever reason, that is a mutation. That could just be the body put the wrong one in by accident, or there was some sort of change due to exposure to like UV radiation, and then the repair process didn't work properly. There's all sorts of things that can lead to mutations. More commonly in bacteria and viruses that replicate much faster than we do. That makes sense, right? So different kinds of mutations. A missense mutation. If we have our AUG and it becomes like an AUC, and instead of coding for methionine, or now coding for alanine, I don't know. But it's a different amino acid we're coding for from that change. That's missense, okay? A silent mutation, if we went from AUG to AUC and it's still encoded methionine and methionine again, like our amino acid didn't change, because there is some overlap in that coding for amino acids due to the wobble effect, which hopefully you guys, that sounds familiar from looking over the lecture and the notes and everything. But anyways, because we have, codons, an amino acid can be coded for by multiple codons, right? That's the wobble effect. It's usually that third position that varies as far as the difference. Anyways, so if you had AUG and it got changed through mutation to AUC, but it's still coded for the same amino acid, methionine and methionine, then that is a silent mutation. It's still coding for the same thing, right? Same amino acid results, same protein essentially in the end. Then we have nonsense mutations. So if you had a change from AUU to UUU, and maybe AUU was phenylalanine, and now UUU is a stop codon that literally signals the bacteria or the cell of whatever kind of cell to stop making protein at that point and disassociate the ribosome. That is called a nonsense mutation because it does not code for a proper amino acid. It just tells it to stop doing its job. So of course, if you had that earlier on than what you were expecting, that can cut, short the entire process of making that protein and can completely destroy the manufacturing of what protein you should have been making in the first place. This is a dangerous mutation to have. Worse than that can be the frame shift mutation. So if I'm telling you, we're reading it as AAU, UUA, AAU, UUA, or whatever the sequence might be on your mRNA, a frame shift mutation either inserts or deletes a nucleotide at some point, and then causes the entire reading frame from then on to shift over by forward one or back one. So now we are no longer reading the same amino acids that we would have been reading. We would no longer be translating out the same protein. So that can completely change the protein, completely destroy production of the protein at that point. So we consider the frame shift mutation to be the most dangerous. Okay, so that is that. So that's genetics in a nutshell, in a real small nutshell. And I hope you guys have watched the lecture. And again, keep in mind that this isn't everything. Anything that's in those lecture slides are fair game. And I will see you guys in the next one. Goodbye.